Hi, everybody. Thank you all so much for being here, and Nick and Garima for the invitation to talk a bit about our work on the subtribe of lichens. And I'm going to start talking about Dictyonematinae, a subtribe of 5 genera. And we can see that there are 2 genera that are filamentous, Cephalastereum and Dictyonema, 2 that are foliose, Corella and Cora, and a microsquamulose genus, Acantholichen. And zooming out a little bit here, they belong to Basidiomycobiont and the order Agaricales and family Hygroforaceae. And this family has the highest concentration of lichenized species in this division. And there are 2 other orders in Basidiomycobiont that have lichens, Lepidostromatales, which is entirely lichenized, and Cantarellales with the genus Multiclavula. We first established genus-level relationships in the subtribe and found Cephalastereum, here in this beige color, is the earliest divergent group and sister to the rest. Then there is a pyrophyletic grade, Dictyonema, representing a transitional morphological and anatomical group composed of these filamentous forms as well as Cephalastereum. Then a derived clade here with 3 monophyletic genera. And these numbers that you see here are the numbers for each genus of how many species we currently have detected. And one thing that kept us wondering, though, was this split here in Cora. And however, because of their uniform morphology and anatomy, we accepted as a single genus. And over time, we were able to establish a much better understanding of the species and their relationships within this subtribe, and then we could move on to the photobiont diversity and their interactions. The photobiont of these basidiolichens are this pretty amazing cyanobacteria genus called Rhizonema, and they're found almost entirely lichenized, but they have also been found in inside pockets of Frulania liverworts. And for me, it's really fascinating how they can have different forms depending on what kind of lichens they're inside. And you'll see that it's more or less round clusters in the foliose forms, and they form these elongated chains in filamentous. And when we're out there in the field, it is not uncommon to see something like this. Many lichens growing together, and we can even hypothesize what's perhaps going on. For example, here, just focusing on the Cyanolichens, we have Parmeliella, likely with no stalk, but then you can also see Coccocarpia and Cora both having Rhizonema, illustrating the potential photobiont sharing between basidio and ascolichens. But inside of the thallus, it is quite difficult to assess photobiont species based only on their morphology and anatomy, and much less their diversity. So we knew a lot about the mycobiont, but not so much about the photobiont and their diversity and patterns. So is it just one? Are there more present? So we really needed DNA sequencing to help us. And in order to assess global diversity of the group and in both myco and photobionts, we did a lot of field work ourselves. And we also studied a lot of herbarium collections and also counted with the help of a lot of collaborators around the world who would send us samples. For the systematics work, I utilized the concept of integrative taxonomy, gathering as much information as possible for each specimen. And for the photobiont work, we're also implying multiple approaches. We usually sequence everything for amplicon, either with high throughput sequencing or with Sanger. So basically everything that we can get ITS for the mycobiont and 16S for the photobiont. And then also for selected specimens, we do metagenomics and culturing. But today I'm going to focus on this more or less new technique in lichens called microfluidics PCR. And in a nutshell, this is a type of PCR based off on this special plate, which in one side you have you can include 48 or up to 96 samples. And then on the other side, you can include 48 primer pairs. And then the machine with this plate does all of the lab work for you. And the microfluidic part is that each of these capillaries will perform a PCR, combining then each one of these primer pairs to each one of these samples. And the magic here consists in barcodes that are present in both the primers and the samples. And with this, then afterwards, you pull everything together, you clean and sequence, and you can then analyze everything with bioinformatics. And I first wanted to find out if there were, I was looking for new loci to help us with species boundaries in this subtribe, but with the lack of genomes in this group, I attempted to use the most phylogenetic informative loci detected in this paper by Dentinger, Gaia, and colleagues for Agaricales, and I utilized the representative of the family Hygrophoracea included. However, it did not work because it was too distantly related. So ultimately, I looked up in the literature and found 19 regions that are commonly found for fungal phylogenetics and adapted this to this technique. And it also created an opportunity then that I was just focusing on the mycobionts to also expand this to cyanobacteria, and it seemed like a very interesting technique to apply for lichens since you can focus in all the different loci of interest. Today, then, I'm going to show, some of these results. First, we perform a network analysis, which help us to better visualize Rhizonema divergence, given that the genetic marker that we are using, 16S, is very conserved, but is still the most widely used for bacteria and the one that works best in Rhizonema in the sense of lab work, what works with PCR. But since networks account for the frequency of each one of these haplotypes, we can quickly see that these larger circles represent that those haplotypes that are more frequent in our sampling. And most of the haplotypes belong to a cluster that included the only named species of Rhizonema up to that point, namely Rhizonema interruptum, but there were two also smaller recognizable clusters that we were recognizing as new species. Rhizonema interruptum is obviously the most diverse and genetically and geographically speaking, but we are also treating this as a species complex that has this sort of blue color, unless it is one of these special or most frequent haplotypes. The same with the other two lineages, you'll see that the same colors are gonna keep coming back. So bear with me. Our current data set has over 1,200 sequences of 16S and is being assembled, but again, is a very conservative gene and there's not a lot of support throughout the tree. Nonetheless, we can still see the two new species clustering together, and usually they have very high support at the species level, but not in the backbone or within those species boundaries. Now reducing to a more manageable size trees, here are the results of some of the loci included in the microfluidics and then combined into a partition concatenated data set. And the colors again depict the three putative species. And we see that while both Neotropicum and Andinum are again monophyletic, interruptum is not. And then we wanted to run a coalescent or species tree in Astro, and all three species then seem monophyletic, showing that both trees, they have some similarities, but also differences between the two approaches, but both of them showing very high support in most branches. We then wanted to see how Rhizonema was placed within a broader cyanobacterial evolution context, and then when it became to diversify. So we reconstructed time-calibrated trees for both the photobionts and the mycobionts. And the inferred stem ages for the three Rhizonema species is between the Middle Jurassic and late Cretaceous, and resolves actually this crown divergence remarkably similar for both markers, and both seem to show that these photobionts appeared in evolution way before the basidiolichens of this tribe that is just very recent and at the bottom here. So these photobionts age, though, they do coincide with the estimations by Nelson and colleagues for the acquisition of cyanobacteria associations in Leccanoromycetes. And then taken out together, it suggests that Rhizonema was first in association with ascolichens and only later with basidiolichens. Next, then, we wanted to see how these photobiont lineages are distributed across the different lichens in Dictyonematinae. And here we created an ITS tree inferred by maximum likelihood, like the one you saw at the beginning. And then what you can see here is the topology of the mycobiont. Then we mapped the different 16S haplotypes into this tree. However, there's just a lot of different colors, and we can then simplify to just show this 3 putative species. First, we can see that as a subtribe, Dictyonematinae has all the lineages, and that all genera are sharing these lineages. So for example, in Neotropicum, you can see some species in Dictyonema, Cantholichen, Cora, Corella and Cora that have those. However, it is a lot more clear now that what was causing this Cora split that I mentioned before is this switch in the photobiont choice in the common ancestor of Cora. And I just wanted to make clear that the main difference between Cora clades A and B, it is this photobiont type and their geography, because Rhizonema andinum is primarily found only in the Andean region and in Central America. And assessing diversity in cyanobacteria is somewhat a challenge still with the markers that we have. But one thing became very clear to us, that despite the very high number of Rhizonema haplotypes, this genus is not nearly as diverse as the mycobionts in Dictyonematinae. Despite all of this diversity, not as much diversity as we see in the photobionts, I still think this cyanobacteria is very extraordinary, because it is the only group of cyanobacteria that has managed to partner with many unrelated groups of fungi, because it is an association with Dictyonematinae, but also in ascolichens of Coccocarpia, Erioderma, Leptogidium, and then also with bryophytes. Lastly, and a more common way to assess specificity is by generating these bipartite networks in which we connect the side of the mycobionts with the photobionts. And if we simplify this image to then just show species that we had 2 or more samples, we can see that most of the mycobionts always associate with one lineage. And there are 2 exceptions of this that will look a little bit close, because there are different examples of how this could happen. And in Cora hexworthiana, you see a very similar pattern than of the genus level, because specimens from Chile and Costa Rica and Colombia, they have andinum, while Brazilian specimens have interruptum. And then we have the example of Dictyonema sericium, which the same species occurs in the same area and still is able to associate with both. But this is extremely rare. And in a more detailed view of the previous slides network, but only now including species that we have 4 or more specimens and haplotypes instead of species only, which further shows how mycobionts are specific in their choice, because before we thought that they were only associating with one species, but here we can see that they're mostly just associating with one haplotype within that species. And the only ones not following these are the ones highlighted here. And okay, I've been talking about the importance of DNA, but this is also something that we can witness in the field. And in this image, we can see both Dictyonema and Cora living together. And this is very much in agreement with all other studies of lichens and their photobiont sharing, that a lot of different fungi may share the same photobionts, and it seems to be something really common in lichens. So as my final message, I just want to say, go out to the field, find what's out there, experience lichens in its natural habitats, but don't forget the importance of herbarium. And for example, combining knowledge of fieldwork, biological collections, and data portals, we were able to detect a new species from Florida in the United States, which might be already potentially extinct, as it was last collected and observed in 1985. So I would like to take the opportunity here to invite you all for our poster on Friday for more work on Cora and species delimitation, including some results of the microbiome part of this microfluidics work. And then finally, we've been advocating for also, fresh specimens, but herbarium has also provided a lot of very useful information combining with morphology and so forth. And we were able to successfully sequence 76% of historical Cora and Corella specimens. And if you're interested in this topic of herbarium importance, please come to Cecile and I's symposium tomorrow, because we have a great lineup of talks. And finishing my presentation here, thanking all of my colleagues as well as multiple funding sources, and I will be happy to take any questions.